I'd also like to introduce my co-host, Brad Day, who is a member of the MPMI editorial board, but he's also a co-author on the work I'm going to be presenting today, and will be moderating questions and probably also answering some of your questions. So,
great. Thanks, Jeanne.
Hi, Brad.
Hi.
Do you want to take a moment to just introduce yourself?
Yeah, I see a few familiar names on there. So my name is Brad Day, and I'm a professor at Michigan State University. And my research is focused on plant microbe interactions, and specifically cell biology of immunity in response to Pseudomonas syringae, and mostly Arabidopsis, but we delve into some real plants every once in a while. Thanks.
Yeah, great. Brad's broad background is one reason why I have asked him to share this time with me. Okay, well, why don't we get started? This is going to be, I'm actually going to be your speaker today because I'm going to be telling you about a project that we've been working on. I'm going to also, if you want to clear up your windows, you can do that so that you can just see the slides. So I'm really excited to be presenting a big project that the MPMI editorial board has been working on for the past year, trying to identify the top 10 unanswered questions in our field in molecular plant microbe interactions. So, and what I'm going to tell you about today is the quest to identify these top 10 questions and what they turned out to be. So I've got my name down here, but I really want to point out that this is a project from the entire editorial board. And people have been involved with it in many different ways. But really, we're focused on this question of plants interacting with microbes. There's a lot we've learned, but there's so much we don't know. So first, as you all know, plants are never alone. They're surrounded by microbes in the air, on their leaves, in the roots, in the soil, and in the water. And these can be all different kinds of microbes: bacteria, fungi, oomycetes. I'm also considering insects and nematodes, and of course viruses, which come via insects. So many different, it's a very complex world, microbial world that they're surrounded by. And they can be at different levels of, different kinds of relationships, different levels of intimacy. So microbes can be just passing through the vicinity. They can live on the surface. They can be attached. They can enter the plant. And some can enter the plant. These bacteria appear to be filling a cell, which is probably dead. But in this image, it's a fungus that's filling a cell, and it's very much not dead. And this is a beneficial interaction, which brings the point that up till now, I haven't really said anything about whether these microbes are good, bad, and neutral. And that's because a lot of that is contextual. And so we're going to look at that today. Okay, so what are the top 10 questions? And why focus on the questions rather than the answers? And as scientists, we know that asking the right questions is key to understanding. So as a grad student, I was told you get what you screen for. And what that really means is that articulating the right question is key to getting the understanding that you want. So, and the flip side of that is by carefully crafting a question, we may be able to gain greater insight into a process than we might have otherwise. So identifying the questions and really taking the time to articulate them is an important process in what we do. As a journal, we usually focus on answers. We publish what we've learned, and that's an important part of what we do. But we realized at MPMI that in order to better serve the community, we also needed to turn our focus forward towards the unknown and ask, what are the big unanswered questions that face us now? So first, why now? And so many reasons. One is we've had enormous advances in our understanding of plant microbe interaction. And I've just pulled out two that were really exciting to me. this ZAR1 resistosome structure, where we're starting to understand at a really, truly molecular structural level aspects of the signaling. And then these extracellular vesicles, which transmit signals from the plant to microbes, from microbes to the plant, in beneficial and pathogenic interactions. Hugely exciting. But so at this point, it's worth asking where we are now. We've answered so many questions. What is still left to ask? And also we've been riding a wave of incredible technical advances in so many different areas, technological, conceptual, different kinds of levels of questions, so many different things. New tools have come out, and these are allowing us to ask questions that we could not have even imagined asking before. And finally, why now? Our planet is facing a rapidly changing climate, and that's paired with an increase in world population. And our population is fed by, well, our food system is based on plants, right? We eat either plants or animals that eat plants, but plants are the foundation of our food system. And this is a hugely important problem that drives us. So we decided it was time to reassess what the big questions are that face us in the field of molecular plant microbe interactions. So great, it's important to ask questions. We need to ask them now, but how do we go about doing that? And first, we discussed it as an editorial board, but we realized in order to really do this properly, we needed to engage the community. And we needed to engage the community on a large scale so that we could reach a diverse audience. And so we did this in many ways. We did this by email, we did this by Twitter, by a podcast that I'll tell you about in a moment, and also by engaging people in person at the IS-MPMI Congress that was held last year in Glasgow, Scotland. And this was important because this was a way that we could reach a diverse audience of people with different backgrounds and perspectives who examine different plant microbe interactions and who ask questions at different levels. And that was the only way we were going to be able to do this. Okay, so the Congress was terrific. There were over 1,400 participants. People came from 52 countries and represented diverse research interests. And you can see us here gathering ideas. Here's Raka Mitra interviewing people for the podcast. People posted questions on a board near the posters. People posed questions in their seminars. Everybody really got into it. There was a lot of discussion throughout the meeting, both in terms of presentations, question and answer, a lot of informal conversations, Twitter, social media, lots of different ways that people engaged. And so I wanted to draw your attention more to our new podcast, the MPMI podcast called Microgreens. Raka Mitra is our host and producer. We've had a few episodes and we are developing some more that will be focusing on these top 10 questions. You can find them at Apple Podcasts or Spotify. Look for Microgreens. So what did we find? And people turned in over 170 questions. It was exciting. It was overwhelming. We managed to narrow them to a core set of 35 questions, which we then posed to the community. And people voted. And about 25%, about a quarter of the people there voted on these questions and helped us to identify which were the ones that were really the burning questions in our field, which were the ones that are motivating our research. So what are they? What are the top 10 unanswered questions in MPMI? And I was excited because in total, these questions were all deeply interesting, timely, and unanswered. So first question is, how can plants simultaneously interact with pathogenic and beneficial microbes. This is a challenge to plants. I've drawn it here in the same cell, but of course, plants are large and they could be interacting in different parts at the same time. So how do they maximize nurture interactions with beneficial microbes while preventing pathogens? So as I told you, plants are surrounded by microbes, which can be good, bad, or neutral. Plants have evolved a series of response pathways to induce defense against pathogens. And it's a complex set of pathways that coincide to change expression, gene expression to turn on defense genes, and also to do things like increase reactive oxygen species as a defense. So what happens if a plant is maximizing, turning on defenses, maximizing them against pathogens? How does that affect interactions with beneficial organisms? Does it inhibit that interaction? And the answer is yes. Plant defenses can inhibit beneficial interactions. And this is a study we did a long time ago, looking at jasmonic acid, which inhibits infection interactions with a number of pathogens, but it can also limit infection by symbionts. So conversely, can the process of interacting with beneficial microbes increase susceptibility to pathogens? And the answer is yes, again. So this is a lovely study by Jeanne from when she was in Gary Stacey's lab, and she's showing that signals from beneficial microbes, both Chitotetros and non-factor, suppress the innate immune response, even in Arabidopsis, which does not interact with those organisms. So signals from beneficial microbes can decrease plant resistance, plant defenses. Okay. But does it need to be, final question here. Can plants discriminate? Can they tell the difference? And that's tricky because here I've given you two examples of microbes, beneficial and pathogenic. In this case, they're both fungi, could have been bacteria. But basically the outside of a fungus is a cell wall made of chitin, whether they're beneficial or pathogenic. It doesn't matter what the nature of that interaction is. They're still made of the same building parts. And so when the plant is detecting things like this chitin surface molecule, which are also called MAMPs, which I'll talk about later, these molecular patterns, beneficial mycorrhizal fungi and pathogenic Magnaportha are both producing chitin, surrounded by chitin, and perceived by chitin. Mycorrhizal fungi also produce these symbiotic signals, lipocyto-oligosaccharides, MEffector, and short chitin oligomers. And in rice, surprisingly, there is one receptor that mediates responses to all of these signals. So clearly, plants are discriminating between the two, but this is not an easy question. How do plants detect them? How can they discriminate? This is still a very active area of research, really a lot of open questions there. And finally, must it always be a trade-off? If we inhibit pathogens, will the plant always lose these beneficial interactions? If they nurture the beneficial interactions, will that always inhibit pathogens? And it's a lot more complicated than that. A number of beneficial microbes can also increase plant resistance to other microbes. And here's an example that came out last year from Hannah Kuhn's lab. Rhizobium nodulation induces systemic resistance against Ericife. And you can see that here in Medicago trincapsula, but also in garden pea, Pisum sativum. And this has been noticed also very strongly for mycorrhizal fungi and probably for many others. So it's not always a trade-off, and they can prime resistance. In this case, they're priming accumulation of salicylic acid, which promotes defenses. Okay, So question number 1 was chosen by 45%, so almost half of the people who voted. Hugely important, on the minds of many. What was next? And the second question was, how does abiotic stress influence plant microbe interactions? Because of course, plants and microbes are interacting not in a vacuum, not in a Petri plate, but normally in the real world where they're subject to all kinds of environmental stress. So classic disease triangle, we know that the environment is one of the 3 nodes affecting that interaction between host and pathogen. So many different ways that the environment can alter whether the plant or the pathogen wins. Humidity, water, temperature, wind, pollution, nutrient availability, many aspects. And I'm just going to show you that it's even one step more complicated. This is a beautiful work from Kenichi Tsuda's lab, which highlights, I think, a lot of the questions that we have not been thinking about, that we don't know. And he showed, his lab showed in this Barron's et al. Paper very nicely that under drought conditions, Arabidopsis has increased susceptibility to a bacterial pathogen. But what's interesting is this was true for old leaves, but not for young leaves. And so I'm going to modify our disease triangle here to have it not just be plant, but also plant development, the developmental status of the part of the plant that's interacting. And that reminds us that it's not just, when we think about this, it's not just on a single cell level, but thinking about the plant as a whole. And finally, here we're thinking about the interaction with a single pathogen. What about that microbiome I told you about? What about all the microbes that are living on the surface of the leaf, for example? And what Barron's showed was that, yes, there's a difference. Each dot represents a different microbial community, by the way, on the leaf. So each dot spot is a different community they're sampling. And you can see here that there's significant differences between the community on salt-stressed leaves versus mock. And this is true for old leaves. It's also true for young leaves, even though we still see a large difference in the microbiome between old and young leaves. So this is telling us that it's a complicated question. We have to consider plant development, but we also have to consider the other microbes in the environment, which was really echoing back to question 1. So the environment also influences interactions with commensal microbes, not just pathogens, and this depends on the developmental status of the plant. Okay, so we know phenomenologically that interactions between plants and microbes are strongly influenced by environmental factors. And while this last study I just told you really dives into mechanism, for most we don't know the mechanism. And we don't know the effect of multiple stresses. And we don't know about stress crosstalk or about the effect of other microbes on this. So this is a huge unanswered area. And as our climate changes and warms, the microbes and plants that are now interacting might not be ones that did in the past. So it's really important that we start to learn something about this question. Okay, abiotic stress, important, chosen by 1/3 of our voters. But the third question, which had almost as many votes, was how do we translate these findings that we're getting in Arabidopsis or other plants to the field? How do we translate these amazing studies and results that we've done in very carefully controlled conditions with model plants? How does that translate to the field? And this is something that people have been thinking about for years and are passionately interested in doing this. translational biology. And I just want to give a shout out to the translational science workshop that's going to be going on tomorrow online. You can register here. And I think Ashley's going to type that into the chat for those of you who want to follow. We have 4 leaders in the field who are going to be talking about how to do exactly that. How can we take these findings? How can we use that to improve our ability to grow crops under complicated field conditions in a way where we can actually help feed the world? Really exciting. I'm really looking forward to this workshop tomorrow and the second translational workshop in January. So keep your eye out for that. Okay. What about 4? Question 4 is, how do microbe-microbe interactions, how do interactions between microbes in the environment affect plant microbe interactions? And this may seem unusually complicated, but remember, the microbiome is unusually complicated. It's composed of many different kinds of organisms. There are thousands of microbes in the environment of the plant. And there's a potential for many binary interactions. Here I've just highlighted interactions between and among bacteria. That could also be happening between and among fungi. And of course, cross-kingdom between those groups. And these interactions can in turn influence the interactions of those microbes with the plant host. So I'm just going to give you one snapshot to highlight how much we don't know. And this is from Scott Gold's lab, really interesting analysis looking at maize seed endophytes. And you can see here they are growing in a petri dish. And what you can, I think, pretty easily see is that this smaller colony, which is SZA, is inhibiting the growth of fusarium, this fusarium species. And they do that by producing essentially antibiotics, which inhibit the growth, but they also change gene expression of virulence genes, thus making this fungus less virulent and affecting its interaction with the plant. So if you were just to study this binary plant microbe interaction, you would be missing the fact that in nature, when these two commonly co-occur, that in fact, this verticilloides is less virulent because of its interaction with another microorganism. So one snapshot that highlights the very complicated world out there, lots of interesting and important questions, and people are really find this a compelling question. Okay, question 5. Question 5 dives into the details, dives into mechanism, and asks whether the way the plant recognizes and responds to microbes, the way we frame a question affects our answers. So just to remind you, I've talked to you a little bit about MAMPs, which are these patterns that are like chitin or aspects of a plant that are conserved, like the flagellum, that are recognized outside the plant versus recognizing effectors and those signaling pathways. So just to remind you, the way that these are framed is the classic zigzag model. And here I've from Jones and Dangle. And it starts off with recognition of MAMPs, which leads to defense responses in the plant, MAMP-triggered immunity. Of course, things keep happening. The life is evolving, and bacteria have evolved, and other microbes, effectors that can block these defense pathways, thus leading to effector-triggered susceptibility. Again, life isn't static, evolution continues. plants have evolved proteins that recognize and inhibit effectors and lead to defense effector-triggered immunity. In this framing of the idea, MAMP-triggered immunity, MTI and ETI, are set as different pathways which do interact to overall provide defense. But it's a very convenient, very nice way for framing these ideas for teaching students. Many of you have probably learned it this way. And the reason I was intrigued to see that this rose to question 5, whether the way we frame this question is something we should revisit. And that comes back to the idea that the way you frame a question influences the answers that you get. And so what they're really saying is, if we framed this idea differently, would we ask different questions? Would we get answers to things we had not previously seen? Okay, and this is just to show you that there are many defense pathways and responses that are actually shared between MTI and PTI. In this diagram, MTI is in blue and ETI in red. And this is the subject of a lovely review by Yulu and Kenichi Tsuda, which is going to be coming out in January in MPMI. It's already available in First Look, so I encourage you to go look at that. They wrote a review focusing on question 5. How is there really a binary distinction? Should we think about this in a different way? And they frame it more from the different kinds of signaling, PRR, NLR, and also ask, how is this machinery used in interactions with commensal microbes? So we think about this as a pathogen machinery, but it's really a machinery for interacting with microbes. Many commensal and beneficial microbes also produce effectors that they secrete into the cell, which also modulate the plant response. So, and the fact that there's so much overlap here, you see lots of areas where the blue and red overlap or intersect or reach similar downstream nodes, suggests that there may not be a binary distinction. Maybe we should be framing this idea in a different way. And if we did, would we ask different questions? So that's a really opened my mind to thinking about the fact that how you ask the question is so important to what you find. Okay. So what is question 6? Question 6 is down here, non-host resistance. I've talked about microbes being beneficial or pathogenic, but in fact, many microbes don't really interact much with the plant, or certainly don't induce disease. They may interact with the plant, but they live as commensals, as we just talked about. So why is that some microbes can cause disease perfectly well on some plants, but not at all on others? And people have been drawn to this question because that really durable resistance that non-host plants have draws them to think, could there be a way that we could transfer this resistance to crop plants? And this is the subject of our very first review on these top 10 questions that has just come out in our November issue of MPMI by Ralph Penstruga and Matthew Mascu. And it's a fascinating review trying to look at this durable, broad spectrum form of resistance and try to think about it less in a phenomenological way and instead bring in mechanism, try to link it to the kinds of pathways I was just talking about in the previous question. And pointing out that resistance and susceptibility are extreme ends of a continuum, and that these intermediate parts are very important and likely illuminating. So can you have cryptic infections? What is the role of the environment? So can the environment take something that's normally a non-host, make it a host? So it's a interesting interplay and a much more nuanced view of non-host resistance than certainly than I was first taught. So in many cases, non-hosts remain non-hosts over long periods and large areas. And this is exciting from a translational point of view, because there's the hope that this may open the door to improve crop resistance to some devastating pathogens. So that's a big area of unanswered questions, exciting opportunity for more research, and hopefully for translation to agriculture. Excellent review. Really excited about this, to have this be the first to highlight. And they will be our next speakers in January. So definitely come join us January 5th for the next talk. It's going to be really interesting. And I should also say that Ralph Panstruga was involved in a lot of the conversations at the conference where we started this. And that's what led him to go more deeply into this whole, what are the top 10 unanswered questions? Okay, moving right along. Question 7, cell death. Cell death, it's a response to a number of pathogens. You get a localized area of cell death at the site of pathogen attack. It's been known for a long time. What is the mechanism? And while we have figured out, there have been many incredible advances recently, including, I'm just highlighting one, lovely work by John Minju and Gigi Chai, looking at the way using structural biology as a tool to examine the way that this NLR resistosome can induce cell death. But even with that, there are still many questions raised. And that reminds me the thing that you all know, which is that for every answer you get a lot more questions. So for every step forward, huge advances like this, we still generate a lot more questions. How is cell death actually triggered? And that was question number 7, still out of the 170 questions initially asked, this still rose very high to the top. Related to this question is question 8. Why do some pathogens have so few effectors and some have so many? So there are pathogens that have 1 or 2 effectors, and there are others, some Pythium species can have thousands. So how does this happen? Why so many or so few? How do you get so many or so few? What's the purpose? What's the role? And that's a question thinking about evolution, rapid evolution, slow evolution, thinking about nuanced interactions. Also, this whole idea of convergence. Remember, there's also exchange of genetic information between pathogens, but yet there must be some benefit to having a lot for certain pathogens, and others can be extremely effective with a few. And that, I'd love to tell you more about it, but this is a big question. This is a huge unanswered question, and very curious one. Okay. And this is closely related to the evolution of virulence. How do microbes evolve virulence? Conversely, how do they lose virulence? And that's related to this idea of number of effectors. I was going to give you one snapshot from Regina Common's lab. Lovely work looking at evolution of novel virulence activities. And the example here is Ustilago matus, corn smut. And you can see here with related fungal pathogens, they both have effectors, these little triangles, that target different proteins that are all the result of ancient gene duplications, right? There's TTK1, 2, and 3. These are paralogs that exist in the same genome. That effectors that originally interacted with, say, kinase 2 or kinase 3, in Ustilago have evolved the ability to interact with a very closely related paralog and make it active as opposed to inactive. And that can make all the difference in some of the phenotypes that these pathogens can elicit in the host. So this is just a glimpse of the fact that you can have gene duplication in the host plant. Gene duplication can occur in the pathogen, and then also evolution, slight changes here in the target that it interacts with and the way that it interacts with the target. And I imagine that there will be many, many different interactions there. And we have another review in process on this one. I'm really excited to see that one coming out soon. So this is How Do You Teach an Old Fungus a New Trick? It's a great paper if you want to dive more deeply into this question. And finally, question 10. How do plant microbe interactions work in an ecological context? And this is a really big question, and it ties together much of what we've been talking about. So obviously, if it's ecological context, one of the big things you have to include is the actual physical environment and any stress that might be there, but also just the physical environment. Other microbes the plant is also interacting with, microbe interactions, and how does this work in the field? So these are all of these, our initial 4 questions actually are encompassed in this final question, which is, if we think about just looking at a binary plant microbe interaction, maybe this one, maybe that one, how are those changed when you add in these other components of the environment? And finally, the other piece here is interaction with other plants, which will be in nature. Of course, it could be very complex like this, but it could also, within an agricultural setting. And so this is very complicated question that is going to lead to a much better understanding, I think, of our ability to translate questions to the field, and also a much better understanding of ways in which our natural environments function and may have implications for restoration or conservation of different environments. Okay, so I appreciate your listening to our quest for the top 10. MPMI would like to be the place where our community discusses the top questions that face us now. We are, in addition to this top 10, identifying the top 10 questions, which is the focus of an editorial that the MPMI editorial board just wrote, which I just talked to you about, laying out the top 10. We're also publishing a series of reviews and perspectives, one for each question. Finally, we're having podcasts. We're developing some more with interviews and discussions that will accompany the articles and explore these ideas. And of course, our virtual seminars, which you are at today. And that's a chance for us to interact with you about the questions that you're thinking about, that we're thinking about, a way to engage and exchange ideas outside the pages of the journal. And this is just to remind you that with these 2 pink boxes, that we have our next review on non-host resistance. Our first review has come out. It's in this month's MPMI. It's going to be our next talk. Really excited about that one. And question 5 is present in the early view and is hopefully going to be the second talk of the new year. Excited to have that. And finally, this was not just me, this was the entire editorial board. People worked on this in many different ways, helping to come up with the initial question, helping at the conference, so many things, going through, culling the questions, figuring out how to disseminate the idea, and finally in writing this editorial that I just talked to you about. So a really terrific editorial board, reflecting, I think, the diverse international community that MPMI is a part of. So I'm going to stop there, and I would love to answer any questions that you have.
We have a couple of questions popping up in the box. One from Morgan Helene from the US. So I'm sure a lot of you have seen the story yesterday on DeepMind on artificial intelligence and the use of AI to predict protein structure. How do you think this might be applied to pursue some of the questions, some of our top 10 questions, and more broadly, help us elucidate some of the functions and ways to improve or enhance plants to resist pathogen infection?
So, great question. I was really excited to see that AI with protein structure. I can imagine so many ways that could be involved in answering the top 10. Effector evolution is obviously huge, looking at these slight differences between effectors, between different strains of the same species. Looking at also with genetic diversity, we're now starting to look at these genetic diversity panels of plants. I've been really excited about using the Medicago ones with hundreds, 400 accessions from all over the Mediterranean, which have slight differences, the same species, but slight differences. Could we use this AI to take the genetic information we have to predict slight changes in protein structure that might help us identify changes in receptors, changes in signaling and signaling receptors and signaling molecules? Super excited about that. I'm sure there's more. I mean, Brad, what do you think about this one? This is such a great question.
Yeah, no, I think one of the real powerful opportunities that lies in front of us is using AI to predict how pathogens will adapt not only to their host, but to climate, and, unveil some of the strategies, more so, I guess, maybe from an evolutionary standpoint. Could we use AI to run an artificial intelligence evolutionary experiment to predict all of the various computations that virulence proteins might adapt or adopt to cause disease? I think this is really exciting, and let alone, just as we move through solving the structures of some of the receptors, some of the R genes or R proteins, excuse me. I think this is going to be a great opportunity for, this kind of next generation. I don't want to say old dog, new trick kind of things, but, for some of those in our audience today to start thinking about how to learn some of these techniques, I think it's going to be very useful for our community.
I think that also highlights the importance of collaboration. You cannot be an expert in everything, and collaborating with people who develop these tools could really enhance our ability to ask these questions.
Right. We have another question here from Banani Mondal from the Bose Institute in India. He said, Jeanne, as you've shown that beneficial and pathogenic fungi are recognized by single receptors in rice, is this common in plants, or are there generally different receptors for beneficial and pathogenic microbes?
Great question. So the legumes, which I know more about, there's been a lot of gene duplication, and generally the receptors have specialized into being more pathogenic versus symbiotic. But even there, there are examples where it's shared. So I think the answer is that there are going to be different strategies that have been adopted in different plants, and also just chance. Loss of something, loss of a receptor leads to change in specificity of the other. And we see that happening in tomato, which originally had two.
Yep.
Great question.
I think one of the other questions that this brings up is, what kind of competition for signaling does a beneficial and a pathogenic microbe kind of converge on in the host? For example, do pathogens, one of their strategies, could it be to occupy a receptor or a signaling event and kind of block that beneficial microbe from utilizing that process, or block the plant from activating other processes that might be of a resistance nature?
Yeah, absolutely. And that's if you think of this interaction on the surface of the leaf or the surface of the root as being this bit of a race between the microbes to see who can get nutrients, basically carbohydrates, out of the plant. And one thing that comes to mind is some of the, I'm forgetting which pathogen it is, but which secretes a chitin binding protein. What does that do? It binds chitins so that the chitin sensing receptors have nothing to bind. That's not activating defenses. So that's a lovely example of it. I'm sure there's many other things in there. I do also wonder if some of the extracellular effectors might influence the binding of these signal recognition events outside the plant to modify the nature of the signal or how well it signals. So that's another possibility.
Okay, we've got another question here, two more. One is from Anonymous. I see that person a lot in these chats. Do you think there are any opportunities for using beneficial microbes in space? For example, introducing beneficial microorganisms for cultivation of crops on Mars?
Great question. Personally, I think that if you want to cultivate crops anywhere, you're going to cultivate them better with microbes. Absolutely. I think plants have evolved to live with microbes. We originally thought of plant microbe interactions as being always disease-causing, aberrant, something that should be avoided. And now we know that plants live in a world filled with microbes. This is their normal environment. This is the way they evolved to live. And I think if you really want plants to be as productive as they can, you're going to give them microbes to interact with. So yeah, however, I would be very careful about which microbes I took with me to Mars. I'm trying to remember the name of that movie where he's planting potatoes on Mars, but
Martian.
Yes, Martian, thank you. And every good plant biologist should watch that movie. But yeah, this question of what gets brought along and what gets inadvertently brought along.
Yeah, so just somewhat related to this, kind of a funny story. When I was living in DC working for the NSF, I had one of my neighbors is a space attorney, and fascinating, and all of the politics that go into space. And so if you remember a few years ago, I believe an Israeli scientist contaminated one of their spacecrafts that they were going to crash land into the moon with a tardigrade. And that was an international incident. So introduction of foreign microbes onto planets, we have to get special permission for that. But space attorney, I mean, that would be a great business card to carry around.
I didn't even know there were space attorneys.
There are, yeah.
There's a lawyer for everything, Brad. So
there is.
Yeah.
Be careful. So another question from Parnashri Day. His question is, endophytes, which are used as biocontrol agents, can be converted into pathogens depending on the environmental factors. Can we really use this basic research at the field level?
That is a terrific question. Yes, I mean, I have so many questions about inoculating with beneficial microbes just to begin with, which is that you're kind of adding, it's like, will it be an invasive species, et cetera? You're really changing the local set of microbes. Of course, for an agricultural field, it's already a human designed environment. So human controlled environment. And I think that there's a huge amount of horizontal gene exchange between microbes. And I would imagine that's a possibility. What do you think, Brad?
Yeah, I think that brings into the question, into the conversation, the role of the environment in really driving when you've got and microorganisms that simply really it's the difference between colonization versus infection. My lab just had a paper published in that area, and some of the reviewers rightly pointed out that is it really infection if it's not a pathogen that's causing disease? So we reverted to saying this is a microbe that colonizes the plant, doesn't infect the plant. So I think looking at biocontrol agents, I hate to say it, but I suspect that with climate change and the other stressors, the other abiotic stresses that we see in agricultural settings, I fear that we're going to see a lot of biocontrol agents that have been quite successful, but yet we don't understand their mechanism. They're going to fail before we really get started in this area. So I think integrating, as you mentioned, Jeanne, some of the influence of environment on all of these interactions and the processes is going to be very important moving forward. Really the convergence of biotic and abiotic stress and the growth defense paradigm.
Yeah. And actually, we're so excited about that question that that's going to be our next focus issue. So keep an eye out for that call for papers soon.
Right.
Yeah, go ahead.
Another question here from Sabine Zimmerman, complimenting you on your illustrations, and says, I completely agree with question 1. How can the plant decide whether it will open the door for symbionts or fight against pathogens?
Yeah, I think it's just such a compelling question. It's something I am personally very interested in. And I will tell you that when I was first writing that editorial, that section got so big that my readers, including Brad, reminded me that there were other questions. Because it's a huge question. How do they do that? And one thing to remember is that often we think about interactions mechanistically at a single cell level, but in fact, the plant is a large organism. And I wonder whether some of these differences they're going to be spatial differences too. Plants are going to be interacting with the pathogen here. One leaf over, they're going to be interacting with something beneficial. And I think, or in the roots, I think it's a huge question, and how that's all coordinated by the plant and balanced, right? So that they can have a normal life. That's a real challenge.
Yeah. I think that also gets into the kind of the underlying way that plants communicate within themselves, how the cells communicate, how they're sending a friendly or a help signal or a danger signal versus open your doors, we've got a rhizobium on our step. So lots of interesting possibilities.
Yeah, and especially these questions of where you have rhizobium in the root system or mycorrhizal fungi, and suddenly you change the interactions with microbes in the leaves, with insects, with fungi. I mean, how does that even happen? So, I mean, I know people are working on it, but there's a lot of open questions there.
Right. More questions coming in. Is it possible, this is from Olalua Ajayi, and I apologize if I've slaughtered your name, my apologies. Is it possible that the fact that the presence of increased beneficial microbes increase plant susceptibility to pathogens is a result of the beneficial microbes inhabiting on the living tissue or the plants themselves, and therefore giving increased access for pathogens to the plant? So, the question would be, does a plant that's undergoing a beneficial interaction, is it kind of letting its guard down? And that as an opportunity for a pathogen to come in, maybe through open stomata or otherwise a dampened immune response when a rhizobium affects a root? Do you have a pythium that comes in and takes advantage of that? It's a great question.
Yeah, that is a good question. And also remember, a lot of these beneficial microbes are growth promoting. So they're also inducing the growth of more plant, more shoot, more biomass. And so of course there is more opportunity for interaction that way as well.
Right.
So yeah, definitely it's like the previous question from Sabina. If you turn down defenses to allow in a beneficial microbe, do you have increased susceptibility, or can you crank it back up again once the microbe's in?
Right.
So it's a good question.
Another question here from Yuva Shankar. What is the current state of microbiome re-engineering, synthetic biology really, in agriculture? They see a lot of opportunities, at least as the use of biofertilizers.
Yeah, I mean, I am hugely interested in this whole root microbiome. Lots of people are working on synthetic communities. So trying to, first you find out who's in the real communities, and then you try to reduce it down to a simple set, a minimal set, and see how they work and whether you can add them. But of course, then you add them back into the environment where there are, if you inoculate your plants in a field, there's already microbes there. There's the environment, there's the sun, water. So it's a complicated question. I do think though, that that's in a really important area for us to go in, because it is a lot faster engineering these different microbiomes than perhaps engineering crops.
Yeah, and I'll just throw in a little bit of my reconnaissance that I learned while I was at NSF. I was in the plant biotic interactions panel with Michael Mishkin. Many of us know this, Michael, very well. One thing that I learned was that I see a great opportunity in the community through the panels of reviewers that I interacted with and the program directors there to let's start really focusing on a couple of those individual pathways that a microbe might encode or perform in an ecosystem. I think we need to quickly move beyond the step of simply inventorying what's in the microbiome and start identifying a few processes that are activated within the microbiome and go from there and look at how those processes interact with other microbes or the plants themselves. So just a hint to those people that work in microbiome research out there. Anonymous, asking lots of questions from Anonymous here. Do plants have adapted immunity? So I can take that one.
I was hoping you would, Brad.
Answer's no. They have innate immune systems. And so those are germline encoded receptors, and that's our R proteins or R genes, but no adaptive immunity as of yet. There are alternative ways that plants use to kind of respond to previous infection, but this whole idea, and shout out to Gita Coker, wrote a really nice review a few years ago on kind of this transgenerational immunity. And so I think, plants are very creative. They do a lot of cool things, but they don't quite have adaptive immunity figured out. So that keeps us all in business, right? As plant pathologists.
Exactly.
Another question from Anonymous: could endophytes be an alternative to elucidating beneficial traits as compared to typical genetic breeding strategies?
Okay, so this is something I'm really interested in. And I think also that when you have traditional breeding strategies, what are you selecting for? You think you're selecting for increased shoot biomass, but maybe what you're really doing is selecting for increased interaction with endophytes, with beneficials below ground. So that's just to throw out there that we often don't know that plant microbe interactions, other than explicit resistance to a certain disease, are generally not considered at all in typical breeding projects, and should be. But yes, I think, genetic breeding, genetic engineering of plants is slow. And that's why this idea of, could we inoculate with microbiomes? Could we inoculate with a specific beneficial? Could we inoculate with an endophyte? Would these be able to enhance resistance of particular plants, crops to drought, to biotic stress? yeah, I think people are definitely looking at that, and that's an exciting open area of research.
Right, and kind of a good follow-up question to that, and I think you've probably answered the question, is from Sabaya Chalavendra, excuse me. Does a beneficial epiphyte prime immunity?
So epiphyte, so living on the surface. Well, that kind of goes back to that whole Barron's paper from Kenichi Tsuda's lab, where he shows that young leaves have increased immunity, and they also have a different microbiome on the surface of the leaves. So suggesting that there could easily be a link. He gets into some of the mechanism there, so I encourage you to look at his paper. Yes, so that's the surface. Certainly endophytes can prime immunity. Epiphytes, I think we still have a lot we can learn.
Okay, Dennis asked a question, but it looks like we answered it. So that's the advantage of this little sidebar conversation that we're having. So I'm going to jump down just to keep us on track, because I think we're maybe entering the witching hour for some.
Yeah.
Matthew Blair from Tennessee State University asked, what percentage of bacterial effectors are plant nuclear targeting? And has there been a lot of gene swapping? And is there evidence of a plant virus gene transfer to bacteria? So 3 tough questions there. I could gloss over the first and say, I'm not sure which ones are plant nuclear targeting. I can remember when, some early work when I was a postdoc with Brian Staskowitz, Pam Ronald and a few others showed at that time, the hot thing was showing that your effector went into the nucleus and could be maybe a transcriptional activator or blocking transcription of immune genes. But I don't know in terms of the percentages what that number is. The nucleus is a great place to look because it lights up really bright when you do these localization studies. But I don't know. Do you, Jeanne, know how many percentage-wise?
I really don't, and that's an interesting question. Certainly, there seems to be an effector for everything. And now that we know that the contents of some of those extracellular vesicles are actually RNAs, I suspect a lot of those RNAs, those interfering RNAs, are heading into the nucleus. I would not be at all surprised.
The next one is there's been a lot of gene swapping. I'm going to assume, just because Matthew's not here and my sitting in the chair next to me, that he's referring to maybe amongst pathogens. Is there a lot of swapping? And yeah, there's pathogens, especially the fungi, but also too some really cool work out of Jeff Chang's lab from Morgan State, gram positives. They're really exchanging a lot of information, virulence factors. They're moving around on small plasmids. The role, I'm thinking of Erwinia amylavra, for example, and work from George Sundin showing that these small plasmids can have virulence factors or antibiotic resistance associated with them. So I think, in the community, in the soil, in the rhizosphere, these microbes are begging and borrowing, and it's like a Yankee swap, I think, with in terms of just grabbing whatever genetic information they can to help them adapt. Is there evidence of plant virus gene transfer? Jeanne, do you want to? I'm not sure about that one.
I have no idea. I think that's a good question. There's transfer of so much from bacteria. Well, I can't think of virus. I can think of certainly there's examples where Agrobacterium rhizogenase genes have ended up in the plant genome. There's a lovely example from Nicotiana, which is kind of a cool, and also sweet potato. But virus, I don't know. I'm not as familiar with the virus literature. That's a really good question. But viruses do hop around an awful lot. It's hard to imagine that they haven't.
Yeah, I know that there is some work on symbiotic interactions between bacteria, viruses, and fungi, but I'm not sure about just the bipartite, the virus bacteria interactions there.
Yeah.
Well, we've hit the end of our list here, Jeanne.
Yeah, well, this has been a lot of fun. I've really enjoyed the questions that people have asked, and also fun hearing from people all around the world. I hope that you will take a moment to look at the editorial, where we've had a chance to go a little bit more in depth than I was able to in this talk. And I'd love to hear your comments on that. So as you read it, you can see my email address there. I'm the corresponding author. Shoot me an email. Tell me what you think of it. Tell me if you have any further questions or ideas. That's one of the things we really want to do, is to really engage this conversation further. So, and I am looking forward to our next seminar, where Ralph Panstruga and Matthew Muskew will have a chance to go much more in depth than I did on that question of non-host resistance. So thanks everybody for joining. This has been a great hour. Thanks, Brad, for
Thank you.
co-hosting and providing all that great additional knowledge that I don't have in different areas. That was why it was so much fun writing this with the editorial board. People with all sorts of interests and backgrounds. It was really exciting. So anyway, see you all in January. Send me an email about what you think about the top 10. Bye, everyone.
Thanks, everyone. Bye-bye.